So for this particular lecture, we're going to look at photosynthetic structures. So this is our first one for this unit. So we're just going to look at the parts of the leaf where photosynthesis takes place. So the first one is the veins. The veins is like your vascular system in your body. So the veins is going to carry water, minerals. It's eventually going to be broken down into xylem and phloem, so it could carry food also. So the vein, like I said, two parts. So the xylem, even though there's an XY, is going to make a Z sound, so xylem. So these are tubes, and it's going to run up and down the plant. And the tube, when I compare it to the phloem, the tube of the xylem is larger in diameter than the phloem, and there's usually fewer of them. The second is phloem. So even though it has a pH, it's going to make an F sound, so phloem, food. Now, if I compare the two, like I mentioned, xylem, the tubes are larger than the phloem. The other thing that's different between them is xylem is only going to move water and minerals. Minerals will be like in your fertilizer. So it's going to move that up the plant. So it'll be like you drinking through a straw. Phloem, moving food, is actually going to go up and down. So the up instance would be carrying those minerals, nutrients, fertilizers from the soil up the plant through the roots, stems, and the leaves. Down would be the leaves that make, through the process of photosynthesis, the leaves would make food. In this case, it would be glucose. And it's going to move that food down the plant to either be stored in the stem or it can be stored in the root, just depending on the type of plant. So carbohydrate might be like storing that food in a potato, the root, or a carrot. And then there are different stem vegetables, etc. So these are two different images. The one on the left is an actual section of a tissue. And so this entire area, that is the vein. So you can see the xylem are these two larger areas, kind of looks like a face. And the phloem, you have a lot more of them, but they're smaller cells. And then all of this kind of orange around it is called the bundle sheath. So it'd be like if you pick up a bunch of straws or pins, okay? And so me holding it in my hand is the bundle sheath. And so the straws or the pins would be the xylem and the phloem. Now this image here on the right is an illustration, and you can see many different illustrations, depends on the artist, but for the most part they look very similar. So they call this the vascular bundle. So that is another name for a vein. So don't be thrown because it says vascular bundle. That is the entire thing. So this illustration is a little hard to tell because the cell size look pretty close to one another, and also the number is very close. So I would try to find an image that's a little bit more distinguishing, kind of like this one right here. Okay? So we're going to look at where photosynthesis takes place primarily in the leaf. So when it says a cross section, so here's my leaf. So if I held it up and I would just take a piece out of it and I would look at the side of it. So that's a cross section. So to me, it always looks like, so I've got this top part here and I've got a bottom one. That's like two pieces of bread. And then everything in the middle, this part right here where all the cells, that's the main place where photosynthesis is going to take place. So think of it the meat of a sandwich, meat, mesophyll. Even within the mesophyll, you have two types of cells, and we're going to look at that in just a moment. And you also have the vein running. The veins would be like in grade school, mom, teacher, whatever, dad, and you did those leaf rubbings and you see all the lines in the leaf. So those are all the veins or the vascular bundles. So that's running throughout the leaf. The two types of mesophyll, you'll notice the top one, the cells are more rectangle in shape and stacked really close to one another. And at the bottom, more irregular shape, and there's white space in between them. And that's going to be really important when you start looking at gas exchange, because we need a place for those gases to go, because photosynthesis One is going to produce a particular gas and it's going to need a particular gas to carry out the process. Here's the cross-section of the actual cells. So this part right here, okay, that is the mesophyll. And you can see it's a little more prominent here. These are the rectangles. That's called the palisade. So the mesophyll is made up of two parts. So we've got the palisade, and the second one, I'll pull that up, is the spongy. So I've got palisade are all these blocks, and they're really close, tightly packed. They are always found at the top of the leaf. Now we're going to talk about plants that are grown in this area that has moderate temperatures, not extreme, some water available to them. And you may hear the term C3 plants, trees, shrubs, cool season grasses. You may also hear C4 plants, and it's just the number of carbons. A C4 plant would be like a weed, corn, some of your warm season grasses. And then you may also hear something called CAM, Crassian acid metabolism, and that would be like your cacti or your desert plants. And they all have a special way to go through photosynthesis, just depending on its environment that it lives in. But they all have in common this mesophyll, and that's where photosynthesis is taking place. So here are the cells, the spongy cells that make up the spongy mesophyll or layer. And then in between it, you have all of these white space. That's where the gases are going to go. So we've got gases like carbon dioxide. We have oxygen. Okay. We also have water vapor. Those are really important through the process of photosynthesis. Okay. So you can tell the difference between the spongy and the palisade depending on the type of leaf and where it's located. Now you've probably noticed that the two parts of the mesophyll, there are actually three different terms here. There's a two-part to the term. We have palisade layer. We have, of course, I've been using the word palisade mesophyll, and this other one is called palisade parenchyma. Parenchyma. I personally, for this class, I would probably use the word mesophyll. It tells you the layer where all the photosynthetic material or where it takes place. When you get to college, your professor may use the word parenchyma, but you can use either term, any of the three terms you want. So both palisade and spongy have the choice of those three terms as a second part for labeling. So palisade layer, palisade parenchyma, palisade mesophyll, and then the same for the spongy. So moving on, so we talked about the mesophyll, which is the meat of the sandwich. So let's go back to the bread part, okay? And you also have something called the cuticle. So the epidermis is just like our skin. It's our covering. They're flat cells. They go together. They interlock. There's no air spaces. And we're going to talk about how is those gases getting in and out if this epidermis there's no air spaces. And then on top of that is the cuticle, so a waxy covering. It helps prevent or help reduce water loss. So a way to think of that, the epidermis would be like your lips. And when you put chapstick on, that would be the waxy covering to prevent it from drying out. So in a plant, you have the epidermis, and on top of that, the waxy covering is the cuticle. So here's our image again. So we can see we've already talked about the mesophyll. So the purple cells here, okay, so that is the epidermis, and then the white on top is the cuticle. And again, how you can tell the difference between upper and lower is where is the palisade and where is the spongy. Now we're going to focus on these pores, these openings, because remember we just said that the epidermis doesn't have any openings or air spaces. So we need to get these gases in and out. So plants have devised something very useful that allows it to regulate the gases going in and out. And these kind of go together, okay? But you need to be careful. They're not the same thing. So we have a term here called stomate, or you might see it as stoma or stomata. I usually use the word stomata, and the reason why there's another term called stroma, and we'll get to that in a moment. So if you say stoma or stroma, they sound really similar, and you don't want to mix those up because they're two totally different structures. So I will use the word stomata, but you may see stomate or stoma. That is the actual hole, pore, opening in the leaf, the opening. So that would be like a door, okay, just the opening of a door. On each side of this opening, there are two guard cells. So they're standing guard, how you want to think of that. And then through osmosis, it can either close the door or it can open the door. So it can either close the stomata, which reduces gas exchange, or it can open it up, which allows for gas exchange. And a way, if you're looking at an actual image, because this might be on a test, okay, if you can go like this and it makes like a cat eye, okay, that's actually closed. And when it makes a circle, that's open. So that's how you can tell the difference. It will never, rarely be completely closed. So if it makes like a little cat eye, okay, so that's going to be closed and this is open. So we'll find some images of that and help you in just a moment. So I already talked about stomate, stoma, stomata, and this is the pore or the opening. That's where the gases are going in and out. And the guard cells is on each side, and it's what's going to control the opening and closing. And again, the gases, oxygen, carbon dioxide, and water vapor, all very important for the process of photosynthesis. So this is a little creepy. This is like a reptile, but if you notice, we talked about kind of the slit, okay? So they would actually say this is closed, okay? And this opening here, okay, that's a stomata. So this is one guard cell, there's one and there's two. And this one tends to be more closed than opened, okay? So now we're gonna look at a process where it's actually looking at the evaporation of water from the leaf. It actually has its own name, and it's called transpiration, to transpire. Let me get my other little image in here. So we have all these openings in the leaves. So when it's really hot outside, just like you sweat a lot, plants kind of do the same thing. So when it's really hot, they're going to transpire or evaporation of water from the leaf really fast. So there is a problem that plants face all the time. If it's a really super hot day and there's not a lot of water around, and the rate of transpiration is greater than the water being brought up by the roots, it's going to lose too much water than it can bring up. In that case, the cells are going to shrink and shrivel and the plant is going to wilt. So for example, if it's summertime and you have your potted flowers and you forget to water your plants, if it's really hot out, they're giving off a lot of water vapor because they tend to want to open their stomata for gas exchange so they can do photosynthesis, which the ultimate goal is to make food for itself. When that happens and you don't water your plants, it's going to wilt, and if it continues, it will eventually die. And you notice when you water your plant, it absorbs everything up and it perks back up. So that is a problem that plants tend to struggle with all the time. So most of the plants in this area, the stomata slash guard cells are open during the day, and then they close down at night. Now there are some plants that it just can't do this. It's just so hot during the day, it's no way that it can keep up with the water uptake. And usually these areas, there's not a lot of water to begin with. So these are our desert plants. And so I only mentioned that you have C3, C4, and CAM, Carassian acid metabolism. These are our specialty plants. They do it the other way around. So when we get to our next lecture, we're going to see that photosynthesis can take place 24/7 because there's two parts to it, one that really requires sunlight and the other one it doesn't need sunlight. So these plants that are in the desert, since it would lose all of its water, it's actually going to open the guard cells at night and then close down during the day so it conserves water. So inside we need to go back to our unit on cells. So you probably noticed we talked about palisade and spongy, and you probably noticed there are dots on this. So if we go back to our cell unit, we remember that plants are the only ones that have a structure called chloroplast. Okay? Um, so this is the image, has little dots. And on this side, this is the actual under the microscope. And so all these little green dots inside, those are chloroplasts. It's just packed full of chloroplasts. Each one of these would be a cell. Actually, when you heat these up, you can actually put this under the microscope, and if it's sitting there long enough, the light will cause these chloroplasts to shake, and those are the electrons that are moving, and so it will look like it's vibrating, which is kind of cool under the microscope. So we're going to look inside the chloroplast. What is going on, and where's all this photosynthesis taking place? So we know it's in the leaf, and we know it's in palisade and spongy mesophyll. Now inside that, what's going on? So we're going to keep digging deeper. So we have something called a thylakoid, and grana or granum. So the thylakoid, it looks like a flattened pancake, and this is inside the chloroplast again. Inside this are pigments and enzymes. And it's these pigments that's going to absorb the light that's going to help get photosynthesis going. And then when I stack up these thylakoids, so I have a stack of pancakes. So one of these is a thylakoid. And when I put a group of these together, sometimes you'll hear it called like a stack of coins. So when I put a stack of these together, okay, that's a grana or a granum. Now, there's an advantage to this. If I have all of these chloroplasts, and inside the chloroplast are these thylakoids, light can penetrate inside a thylakoid. I get more surface area if I have stacks of thylakoid and I can penetrate all of them to where photosynthesis is occurring in 5 of these rather than 1 of these. So the key to stacking the thylakoid membranes is to increase surface area to allow maximum absorption of the sunlight. I get more bang for my buck. So this is our chloroplast, and you probably remember this from our cell unit. So we've talked about the thylakoid, and that's one of these little sacs here. And we've talked about the grana, so that would be an entire stack. So you see I've got several stacks. And in just a moment, we're going to get to something, the stroma, okay? I'm not going to ask you to call it the stroma lamella. You may do that at another class. So we'll just talk about stroma, okay? The stroma is this light green going between it, between these stacks of thylakoid membranes or grana, okay? So the part between the thylakoid membranes and the grana is called the stroma. So this is where the dark reactions. So photosynthesis takes place 2 parts. I already kind of mentioned that. We have our light reactions, which require sunlight, and we have our dark reactions that actually uses carbon dioxide. And we know that carbon dioxide is available 24/7. So no matter what time of day photosynthesis is going on in this plant, because it needs to make a lot of food. It needs to make food to store for animals to eat. It also needs to make food for itself to live, grow, and produce if it needs flowers or fruit or etc. Okay, so we've looked at the thylakoids, grana, and stroma. Now, inside these chloroplasts are also something called the pigment. Pigment, and you can always tell when it says fill, okay, we're referring to pigment. When we see the ending plast, we're talking about a structure. So that's how you can tell the difference. And chloro means green. Okay, so chloro means green. So these chloroplasts is going to make chlorophyll. It actually has other pigments too, but we're going to focus on chlorophyll. There's actually 5 different types. There's A, B, C, D, and a 5th one called bacteria chlorophyll that's only found in bacteria. But for most of our plants, we have A and B, okay? Um, the way this works is that um, you have these wavelengths of light, the spectrum. So the primary colors that it's going to absorb: violet, blue, orange, red. So it's going to absorb that, and then it's going to reflect green. So orange could also include the yellows. So we're going to do an activity in class where I'm going to demonstrate this. So basically, wavelengths of light, anything that you see is what it's reflecting. So if you have a red plant, then it's reflecting primarily the pigments that have orange and red in them. So we see this when we look at our accessory pigments, and we'll talk more about that a little bit later. So actually chlorophyll shuts down, these other pigments kind of turn on, and so you have these color changes. And there's things that trigger that also. So we are going to get more into light and dark reactions, but I just want to give you this little study hint that you can kind of help remember light and dark and where they take place. And that's kind of the premise of this whole lecture: where these processes take place and what is it about? What is it producing? What does it need? Products, reactants, etc. So thylakoid has an L in it, and that's where light reactions take place. So light reactions take place in the thylakoids. I can also add the grana here because we have our stacks of thylakoid membranes that make up a grana or granum. The stroma has an R, so that's our dark reactions. So that's using carbon dioxide. There we go. Okay, so let's kind of put all this together, put it in perspective. So we start off with our basic leaf. We're going to look at the cells of a leaf. We've seen that before. And we're just going to move down here. So inside my leaf, I have a chloroplast. Inside the chloroplast, I have my thylakoids, and I can also add grana here. And then we'll get into this in our next lecture, but these membranes, okay, we're going to look at this to where light reactions, remember, thylakoid is where light reactions take place, and we'll also look at the stroma where dark reactions take place. But in these membranes, so this part right here is this line, this membrane. This is where all these electrons, the hydrogen ion, where all of this is going to take place, that we're going to use oxygen and water. We're going to make, in the dark reactions, we're going to make some sugar, some glucose. So for right now, just understand how piecing these structures together. So that ends this first lecture. Wouldn't be a bad idea to go back and look at the main structures. And of course, you have your guided lecture sheet. So any questions, just ask me in class, and I will see you later.